kung ito lang po sana ay may implement not only by BFAR, not only by other regulatory agencies, but also with the assistance of our local government units, I think this is already enough. Ang kailangan lang kasi it's just we have to coordinate and collaborate efforts. The government cannot do it alone, but also the stakeholders have a big part also to do. I mean, kung gagawin talaga ng stakeholder na sinabing hindi pwedeng mag-dump dito, hindi pwedeng mag-produce dyan, hindi pwedeng maggamit ng ganito, I think those problems can be maiiwasan. So it's just for the government to implement and for the stakeholders to comply.
Okay, so thank you. I think this is the last question, but it's a follow-up to what has been discussed. This is from the province of La Union, LGU. LGUs are frontline services providers. Projects are now in place on fishery development. However, budgetary and technical know-how constraints hamper its sustainability. So there is a challenge for NAST. What are the budgetary and technical interventions available from NAST? So I think NAST can give you technical assistance or technical advices, but we don't have any budgetary. NAST is not a funding agency. But I guess the point is, while we do have national programs, the local governments need technical know-how and, of course, the budget to trickle down the activities from the national to the local. So we need to wrap up this session. I would like to thank the speaker, Dr. Palier, and our discussants for very fruitful discussions. We do have some more questions here, and I would like to send this to the NAST Secretariat, again, for their processing and action. Okay. So at this point, may we call on Academician Fabian Dairit, our NAST Acting President, and nandito pa si Director Victor Mariano, to present our certificates and tokens of appreciation to our resource person and discussants, as well as to our rapporteur.
UN Assembly.
Yes, so for Dr. Palier, and our discussant, Ms. Lilian Rueca. Nagpalit kasi ng mga discussants, pero mga bosses wala dito. And also Ms. Mia Baldonado. So can I also call on our rapporteur, Ms. Tess? Okay na. So tapos na po. Okay, ha? right, so that ends our session. So may we remind the audience that you have to present your accomplished evaluation form to the registration area to claim your certificate of attendance. Copies of the plenary session presentations will be available at the NAST website, and we would also like to invite everyone to follow us, the NAST social media accounts on Facebook, Twitter, and Instagram. And we would like to remind everybody that we will resume for the second plenary session, where our speaker will be Dr. Maria Lourdes San Diego Maclon. So thank you very much.
Moderator for this session. We are going to move from one session to another without a break. Welcome again to the second plenary session of the Luzon Regional Scientific Meeting. This session's speaker is Dr. Maria Lourdes San Diego Maclon. She will discuss water quality issues linked to fish production. Dr. San Diego Maclon's presentation will be followed by discussions from Ms. Adelina Santos Borja from Laguna Lake Development Authority and Dr. Rico Angkog from the School of Environmental Science and Management, UP Los Baños. The rapporteur for this session will be Mr. Randolph Valencia, Science Research Specialist 2 at DOST Region 3. Can I ask our speaker, our discussants, and rapporteur to come up to the stage? Dr. Maclon. Papakilala pa lang ang panel natin. Dr. Maria Lourdes San Diego Maclon is a professor at the Marine Science Institute in UP Diliman. Her research interests include nutrient biogeochemistry of coastal system cycling and human well-being in social ecological systems, funded by the Research Institute for Humanity and Nature based in Kyoto, Japan. She leads the LLDA team in an ongoing revision of the zoning and management plan of Laguna de Bay for aquaculture based on the lake's carrying capacity. Ms. Santos Borja has an MS in Biology and in Environmental Science from the University of the Philippines. She specializes in the field of limnology as a UNESCO Fellow at the Institute of Limnology of the Austrian Academy of Sciences. She participated in the training on lake water quality management by the Australian National University in Canberra. She has also completed the first Temasek Foundation Water Leadership Program of the Lee Kuan Yew School of Public Policy at the National University of Singapore. The other discussant will be Dr. Rico Angkog, who is an assistant professor at the School of Environmental Science and Management of UPLB, where he also finished his PhD in 2010, complemented with the Sandwich Program in Nagoya University. Starting in 2013 up to the present, he has served as the managing editor of the Journal of Environmental Science and Management, an ISI index journal based in UPLB. He won the 2009 award for Asian Young Professional on Water Research. Our rapporteur will be Mr. Randolph Valencia, who is a Science Research Specialist 2 at the Department of Science and Technology Regional Office 3 and has been with DOST since 1996. He completed his undergraduate degree in computer science at the AMA Computer University in 1993. He started out as a computer programmer and has moved on to graphic design. Mga reminders dito, we would like to remind our speaker that she'll be given 20 minutes for her presentation, and each of our discussants will have a 15-minute allotment. And then there will be a 10-minute open forum, whatever time we save, of course, makes the open forum longer, right after their presentations. We also want to remind the audience to write down their questions on a piece of paper to be provided by ushers and usherettes, together with your name and affiliation. If you do not put your name, you get lower priority. the ones who identify themselves get read first. Please give your questions to the usher or usherettes. Nasa side sila ng stage. And to manage, already said earlier, the written questions will be given priority. Okay. Okay, can we start off now with, may I ask Dr. Lourdes San Diego to start?
Magandang hapon po. It's an honor to be introduced by our Chancellor Tan. So, the title of my talk is Water Quality Issues Linked to Fish Production. The outline goes as this. So I'll talk about fish production trends, mariculture as a BFAR strategy, mariculture and environment, giving Bolinao and Anda as a case study, way forward, and take-home message. So we are a growing population. This is the population trend in the Philippines. Currently, we are over 100 million people. And we need to feed all these people, and the protein diet of the Filipinos average is 56% of the total animal protein comes from fish. So we have to come up with fish to feed all these people. So what are the sources of fish production? We have aquaculture, which is in short culturing of fish, whether in marine, brackish, and freshwater environment. We also have municipal fishing as a source of fish production, and we also have commercial fishing. Municipal, when you use boats that are less than 3 gross tons, and commercial fishing when you use boats that are bigger than 3 gross tons. If you look at a comparison of all the 3 sources of fish production, aquaculture represents 52% compared to municipal and commercial fishing. And over time, we see that there is a decline in capture fisheries. And the red part of the bar represents the aquaculture production. So there has been an increase over time in aquaculture and decrease in capture fisheries. And the decrease in capture fisheries is due to overfishing, which is brought about by improved efficiency of fishing gears and increasing number of fishers, destructive fishing methods, and pollution and loss of marine habitats. So aquaculture production in the Philippines has been increasing over time. And what are cultured would be number 1, seaweeds, number 2 would be milkfish, number 3, tilapia, and number 4, shrimps. If you look at, so aquaculture can be, is culture in seawater, in brackish water, in freshwater. So the green part of the bars represent marine culture or mariculture. And we see that over time there's been an increase in the size of the green bars. Now focusing on mariculture. So mariculture is farming in coastal areas or marine offshore environment. This is actually being promoted as a livelihood diversification option for coastal communities in the Philippines. So BFAR promoted mariculture for 3 reasons. 1 is to generate employment and alleviate poverty. Number 2 is to promote marine fish culture as an alternative source of livelihood, especially for the marginalized and sustenance fisherfolk. Number 3, develop an area with appropriate equipment and infrastructure that will allow fishermen, farmers, and investors to operate more cost-effectively and securely. And number 4, promote the use of environment-friendly inputs and farm management practices. So the objectives are good, no? But what happened? So if you look at this is from the BFAR website some years back. This is the location of existing mariculture parks and zones.
So a mariculture park is like an industrial park. So it's like you build condominiums, like building condominiums, and you wait for investors to come in. They either rent, or it's not essentially to own, but to rent the structures that they put in, and then to put whatever they want to culture. So over the Philippines, there have been like 50 mariculture parks, mariculture zones established, but I don't know what the current number would be that are existing still. But here it's written down below. You have for launching, you have 9. Okay. So according to a paper written by Salayo et al. In 2012, she evaluated the mariculture project of BIFAR. And 10 years after the launching, which was in 2001, only 0.54% has been established of the total 50,000 hectares that were planned to convert into mariculture parks and mariculture zones. So why is there a very small percent of what's been launched? Number one, lack of thorough feasibility study and environmental impact assessment, and absence of clear agreement between the local government and BIFAR. And number three, unsuitability of some location for development and other inefficiencies found in project implementation. So I'd like to talk about mariculture and the environment. So one issue is you have to put these mariculture parks in the proper areas, not just anywhere in the local government coastal waters. So because there are a lot of environmental effects. So in particular, focusing on water quality issues. So if you look at the list of environmental impacts of aquaculture and mariculture, there are many. No? But I'd like to look at the last one, which is the release of waste. So the release of waste can lead to algal blooms. They can be toxic or non-toxic, depletion of oxygen, fish kills, and damage to ecosystems. So let's go back to this map, and I'll talk about the case of Bolinao. It wasn't declared as a mariculture park, but we've had efforts by investors to put up fish pens and cages in the coastal area. So the study area is in the northwestern part of the Philippines. This is Bolinao, and I also include the adjacent local government, which is Anda. So what is common between Bolinao and Anda? So they both share common waters. They both engage in fish farming. So this is Bolinao and that's Anda. And then they culture bangus, chanos in fish pens and cages. And they both experience algal blooms and fish kills. Okay. So I'd like to talk about the environmental impacts that were listed a while ago, and first focus on the first three. So we have a marine station at Bolinao. Now it's called the Bolinao Marine Lab, and we've had opportunities to study the waters there, and we've seen the emergence of mariculture in the area. So we're able to monitor the changes over time. And this is the HAB, harmful algal blooms. So if you look at the timeline of occurrences of these blooms in Pangasinan, there have been a lot already. And the major bloom from which this study started or emanated occurred in 2001. So that's the one encircled, no? This bloom caused a lot of fish kills that resulted in about 600 million peso loss to the local government of Bolinao. So after that, there have been several blooms of different types of species of dinoflagellates, whether toxic or non-toxic. And in addition to that, there have been also episodes of fish kills. So we'll try to look at why this is happening and why it is happening still until now. So let's begin. So this is a case study to link the expansion of fish farming, the existence of eutrophic waters as a result, algal blooms, and then fish kills. So number one, why fish kill happen? There are too many fish pens and cages. So on the left is the coastal development plan for Bolinao. So this coastal development plan is prescribed by the national government for the local government to have. So these are what you would do to your local waters. You will set aside areas for culture, you set aside areas for navigation or for recreation. No? So on the right are, So in the Coastal Development Plan, the symbols represent the symbols represent where you can put the recommendation and where you can put the fish pens and cages. No, so you cannot put it anywhere you want. So this is recommended, and there's a carrying capacity for the number of structures that you will put. So the carrying capacity that they have recommended is only 544 structures. This carrying capacity number is based on hydrodynamic modeling. So they determined the number of structures using friction points. So the friction points is where the water will slow down. So if you have a structure, the water will definitely slow down as it passes through the structure. On the right figure is the data for 2007 on the structures that they put. You'll notice that not only in the prescribed areas, but they have already expanded. So the red represents fish cages, the green represents, the blue represents fish pens. No? So dumami na talaga ng todo. Okay, this is the data from 1995 to 2005 of the number of structures. Again, the carrying capacity should only be 544 structures. But in 2001, when the major fish kill event occurred, the number of structures doubled. So it went up to more than 1,000. After that, so actually we were warning the local government, Bolinao, na magkakaroon kayo ng fish kill kasi sobra-sobra na. You have to lessen and you have to regulate, no? Pero hindi sila nakinig until the disaster happened. After which, they complied, no? If you notice, after 2001, bumaba na yung bars. So, kinontian na nila, nag-comply na sila sa 544 carrying capacity. So, ano ba nangyayari pag nagkaroon ng fish pen and fish cage? Yung nasa left is a speed data, speed of the water. Ito yung paano kabilis water flows, no? If it passes through a fish pen, the right side is if it passes through a fish cage, no? So in general, what this says, the red bars represent speed of water outside the pen. Siyempre, mas mabilis sa outside, no? Yung hindi na disintegrate rapidly. They want intact feed. So, but however, based on experiments that were done, if we put the feed in a petri dish and just let it dissolve, so the stagnation rate is very fast. So ito yung in a petri dish. Can you imagine if it's given in the fish pen or fish cages? So the water is very agitated. So very fast yung disintegration ng feed. So it takes less than a minute for the feed. So what happens to the crumbled feed or the disintegrated feed? It just ends up as feed waste. So milkfish feeds are very unstable in water. And so if it dissolves, so yung nutrients na release sa tubig. Okay? So this is just a balance, nutrient balance, no? Nitrogen, 50% assimilated, and the rest are released or excreted, and phosphorus, 23% na yung assimilated. So malaki yung wasted, no? Wasted material. Okay. This is a study we did to look at if we put feeds in a beaker, how much nutrients, how much nitrogen, and how much phosphorus is released in a period of 1, 2, 3, 4 days. So the bars are increasing, and if you look at the tallest bar, it means it's written there 850%. So nitrogen in the form of ammonia, after 4 days, yung content ng nitrogen sa tubig would increase by 850%. Phosphorus by 180%. That's a lot of nitrogen and phosphorus that you're putting into the water. Okay? We're fortunate to be in Bolinao, so we have a long-term data, no? So dito lang pinapakita ng the left top graph bars, this is the number of structures over time. So mabait pa rin yung Bolinao. They kept the number of structures below the carrying capacity, which is good, no? And however, if you look at the nitrate, tumaas na siya. If you look at the phosphorus, tumataas siya over time. If you look at the ammonia, tumataas. And then if you look at the chlorophyll, which is an indicator of algal biomass, the higher the chlorophyll, the more plankton there are in the water. Okay? Alright, hindi lang sa tubig, dun din sa sediment. So under a fish cage, you will see the sediment as black. Black, stinky because of sulfide being released. No? On the right is a sediment away from the fish cage. What you want would be the brown sediment. It means it's oxidized, unlike the very reduced sediment on the left. And if you look at above the sediment, merong puti-puting parang bulak. That's not that's a bacteria. It's called vegetaua. It's a sulfur-loving bacteria. So number 3, eutrophic conditions causing algal bloom. So ang dami-daming food that's released into water column, and during the major fish kill in 2001, the bars represent how much dinoflagellate is a kind of plankton, no? Are found in the area during those days. If you look at January 31 and February 1, the bar is completely filled. It means 99% of what you find in the water is made of that organism you see on the right bottom corner. So it's called Porocentrum minimum. Okay, and this is the scenario during the fish kill. So you have a brown bloom, and unfortunately, so over time, nagkakaroon ng bloom. So this is just to show you na within a period of a month, iba yung nag-bloom. For example, May, nag-bloom a species called Alexandrium. In June, nag-bloom Skeletonema. So within a month, there will be a change in composition. And buti na lang, Alexandrium, Skeletonema is non-toxic, Alexandrium is toxic. So you get a combination of toxic and non-toxic blooms. Okay, number 4, depletion in dissolved oxygen. So pag namatay yung blooms, you use up all the oxygen, and then what happens? There will be no oxygen left for the fish. So the fish will die. Okay, this is oxygen. According to DNR, oxygen should not be less than 5 milligrams per liter. But during the bloom, which is 2002, oxygen levels went all the way to 1, and sometimes 0 for some cases. Oh, this is an experiment. So we put, again, feeds in a beaker, and the top graph says that over 24 hours, the y-axis is oxygen. So oxygen over 24 hours decreases to 0. So ang ganon kabilis, if feed is wasted, the amount of feed wasted is 85%. However, if the amount of feed wasted is only 30%, hindi bumabagsak yung oxygen. So this tells us that the composition processes would really consume all the oxygen. So this is a summary of why the fish kill happened. Okay? And this is a picture of the fish kill. So the white images there are all bangus floating, and the fish kills are not only happen for the bangus but also for the reef fishes. Okay. So the right figure is a map with the Mariculture Park. The left figure is a map where you have red tide blooms and all other types of blooms. So it's interesting to see na parang yung location kung saan nilagyan ng Mariculture Parks, parang doon din nagkakaroon ng bloom. So what happened in Bolinao can happen in the other sites. So medyo scary, no? So there's also damage to ecosystem. So if you look at Bolinao, the fish cages are the red marks. The green are your seagrasses, and the purple are your corals. So the question is, yung effluent pagaling sa mariculture activities, do they reach the coral area or the seagrass area? So the size of these are stations from the left. The blue dots represent coral stations. The green dots represent seagrass stations, and the red dots represent the stations where you have the fish pens and cages. So the bigger the dots, the higher the concentration. Yun lang yung ibig sabihin nito. If you look at the size of the dots, mataas yung nitrate and phosphate doon sa mariculture. So malaki yung release galing sa mariculture activities. And the pH is also very high. Sorry, very low, acidic. There's very acidic conditions. So you're creating an acidic environment, a very nutritious environment. Okay? There was modeling done to look at the amount of material, or to look at where this waste goes. So the X represent release points. Imagine niyo yung X, doon nandun yung mga fish pens and cages. So after 4 days, so yung seagrass and let's see if the release material ends up in the seagrass and mangrove and coral area. So after 7 days, you see it's and after 10 days, so it's reached the seagrass and coral area. So the ecosystems are affected by your effluents. Okay, that is during the wet season. During the dry season, hindi masyadong malaki yung effect. Alright, so yung seagrass, the seagrass photo on the left is a very nice seagrass area. And then as you go to the right, you have a very turbid and yung medyo puno na ng algae na mamatay na. So that's what you can experience when you have fish pens and cages above your seagrass. So it's very, very critical to not put your structures where you have your ecosystem habitats are. This is a change in seagrass over time. And we also looked at nutrient gradient of nutrients. So from a site that's very high in nutrient to a site that's very relatively clean. No? So we looked at coral species and we actually put them there and see whether they survive. So the y-axis is survival, the x-axis are 3 points, the reference, the intermediate, and the high nutrient area. So the reference station had the highest survival in the Okay, so the question is, carrying capacity is followed by Bolinao, but however, meron pa rin fish kill. So, but why do fish kills still occur even if carrying capacity is followed by Bolinao? The answer is, kasi it's not established in ANDA. So there's no limit for ANDA, and remember they have common waters, they share the waters. So if Bolinao is complying, but ANDA is very happy and putting in more structures, then there's nothing Bolinao can do about it, no? So you have to establish the carrying capacity for ANDA, and you need to improve the feeding practice, okay? So this is just a model again to look at dissolved oxygen. So the red represents high oxygen, and the blue represents low oxygen. So on the left is keeping the present feed amount. So you have areas na medyo mataas yung oxygen and areas na mababa na yung oxygen. On the right is reduce the feeding amount by 25%. This is just saying that dumami yung areas na naging red, no? So mas kumonte yung areas na remain as blue. So if the model says that if you reduce the feeds, then oxygen levels will improve. Okay? Okay, this naman is another model